To understand bioinformatics, we first need to think about the information contained inside our cells. Inside the nucleus of most human cells are chromosomes. These chromosomes are made of DNA, the famous double helix. And DNA itself can essentially be represented using just 4 letters: A, C, G, and T, which correspond to the 4 chemical bases. The particular order of these letters forms the biological instructions that help cells develop, function, and reproduce. So at one level, biology can actually be thought of as an enormous information problem. For example, one copy of the human genome contains roughly 3 billion DNA letters. 3 billion! If we tried to print the entire sequence as ordinary text, we would end up with an extraordinary amount of information. And that's just one person. Today, researchers can collect biological samples from very large numbers of people and use high throughput sequencing technologies to read their DNA. A blood or saliva sample can ultimately become digital genomic information. So think about what is happening here. On one side, we have people, then we have biological samples, which go through sequencing machines, and out the other side comes enormous amounts of digital data, DNA sequences, genetic variants, genome-wide measurements, and other information that researchers can analyze. Modern genomics has therefore created something biology did not have at anything like this scale a few decades ago: big data. And the larger our studies become, the more important computational analysis becomes. And humans aren't the only organisms we sequence. Different organisms have very different genome sizes. Some microorganisms have relatively small genomes. Plants, parasites, model organisms, and humans can have much larger ones. And researchers routinely sequence not only humans, but also pathogens, animals, plants, and entire communities of microorganisms. So the challenge isn't simply that we have one enormous human genome, it is that modern biology is producing vast quantities of information across almost every branch of life. Genomes, RNA, proteins, microorganisms, clinical data, images, and much more. We therefore need a discipline capable of making sense of all of it. That discipline is bioinformatics. At its simplest form, bioinformatics is the use of computational approaches to organize, analyze, and interpret biological data. It isn't purely biology, it isn't purely computer science, and it isn't purely statistics. It combines all three. First, biology gives us the questions. For example, which genes are involved in a disease? How does a pathogen evolve? Why does one person respond differently to a medicine compared to another person? How are two populations genetically related? Or how are two individuals genetically related? Then we have computing. Computers allow us to store enormous data sets, and develop algorithms and software capable of processing them. And finally, we have statistics. Statistics allows us to determine whether an apparent pattern is likely to be meaningful. So bioinformatics brings these areas together, and increasingly we can add fields such as artificial intelligence and machine learning to this picture. So it's almost impossible to talk about bioinformatics without talking about the omics first. So this includes studies in genomics, and genomics includes whole genome sequencing, whole exome sequencing, targeted sequencing, or genotyping specific positions across the genome. Depending on the question, we might analyze millions of genetic variants from thousands of individuals. Next is transcriptomics. So while DNA tells us what genetic information is present, we know that genes are not all equally active in every cell. So the transcriptomics allows us to study RNA and therefore examine which genes are being expressed. We can analyze RNA from an entire sample, look at RNA in individual cells using single-cell technologies, or even investigate where particular genes are being expressed within a tissue. So already we are moving beyond simply asking, what genes does this person have? We can also ask which genes are active, in which cells, and under what conditions. Then we have epigenomics. So we know that the DNA sequence itself is only part of the story. Cells also regulate how DNA is packaged and which genes can be switched on or off. Epigenomic technologies investigate these regulatory layers. So we can study DNA, methylation, chromatin accessibility, and the interactions between DNA and regulatory proteins. This helps us understand why two cells with essentially the same DNA can behave completely differently. For example, a neuron and a liver cell contain largely the same genome, but they use that genome in very different ways. Next comes proteomics. So genes can be transcribed into RNA, and many of those RNA molecules are then used to produce proteins. Proteins do much of the actual work inside cells. Proteomics therefore attempts to identify and measure proteins on a large scale. Again, this produces substantial quantities of data, and again, bioinformatics is needed to process and interpret that data. Then we reach metabolomics. So metabolites are small molecules produced or used during biological processes. By measuring many metabolites at the same time, researchers can gain a snapshot of what is happening inside a cell, tissue, or organism from a metabolic perspective. Related approaches include lipidomics, which focuses specifically on lipids. And then we have the microbiome and metagenomics. So humans don't exist biologically in isolation. Our bodies contain enormous communities of microorganisms. Rather than growing each microorganism individually in a lab, researchers can sequence genetic material directly from a sample. So for example, to study the gut microbiome, we can sequence a stool sample. So bioinformatics can then help determine which organisms are present, how abundant they are, and potentially what biological functions they are carrying out. The same principle is used in environmental samples. A sample of soil, seawater, or wastewater can contain genetic information from huge numbers of organisms. So, now put all of these technologies together: genomics, transcriptomics, epigenomics, proteomics, metabolomics, and microbiome data, and increasingly, measurements at the level of individual cells. This is where modern biology becomes incredibly powerful, but also incredibly complex. Imagine studying a patient and having information about their DNA, gene expression, proteins, metabolites, microbiome, clinical measurements, and treatment history. Each dataset tells us something different, but the really interesting discoveries may come from connecting them, and this is what we refer to as multi-omics. Integrating these different layers of information is one of the major challenges and, of course, opportunities in modern bioinformatics. So, let's take a look at what you can use bioinformatics for in the grand scheme of things. The answer is an enormous range of biological questions. So, we've already talked about human health, where bioinformatics can help researchers identify disease-associated genetic variants, discover biomarkers, and investigate why individuals respond differently to treatments. Then in pathogen genomics, we can sequence viruses, bacteria, and parasites to study transmission, outbreaks, evolution, and drug resistance. In evolutionary biology, genomic data allows us to reconstruct relationships between organisms and populations and investigate how species have changed over time. In protein science, computational approaches can help us analyze protein sequences, predict structures, and investigate the effects of mutations or variants. In microbiome research, bioinformatics helps us characterize the complex microbial communities living in humans, animals, and the environment. Then in agriculture, genomic information can support crop and livestock breeding, disease resistance, and the development of more resilient varieties. And these are only a few examples. Bioinformatics is now involved in almost every area in which large amounts of biological data are being generated. This is one of the most exciting things about the field. Genomics is no longer something that belongs only inside specialized labs. It increasingly affects questions that matter directly to society, and it is at the heart of precision medicine. Bioinformatics provides the bridge between the enormous amount of biological information we can now generate and the knowledge we hope to gain from it. If you enjoyed this video, subscribe for more explainer bioinformatics and genomics videos, and let us know in the comments if you have any questions on this exciting field.